Hello, I'm Wassilios Meissner, and I will speak about what is new in immunotherapy. Indeed, immunotherapy is currently the most advanced in clinical development for treating Parkinson's disease, but there are other approaches to tackle alpha-synuclein, and this is what I would like to illustrate in this introduction here on the left side. As you can see, there are different ways to tackle alpha-synuclein, the aggregation and the accumulation in Parkinson's disease. One way could be to reduce the genetic expression of the SNCA gene that encodes for alpha-synuclein. That may be particularly interesting and relevant for patients with duplications and triplications of the SNCA gene, where the concentrations of alpha-synuclein is increased by twofold or threefold in brain cells. Another way could be to enhance the clearance. We know that alpha-synuclein is metabolized through autophagy and proteasomal function, and we know that both have deficits in Parkinson's disease and may thereby contribute to the accumulation and aggregation of alpha-synuclein. There have been proof of concept studies and preclinical models that have shown that indeed with these approaches you can obtain beneficial effects on the neurodegenerative process and the aggregation of alpha-synuclein. Another way could be to target directly the aggregation. There have been a few small molecules recently developed and validated in preclinical models where the compound is interfering with the aggregation, for example, with the dimerization, and by that means reducing the aggregation and accumulation of alpha-synuclein within the cells. Another way could be to act directly on post-translational modifications. Post-translational modifications are frequent. So the alpha-synuclein protein is undergoing frequent post-translational modifications, phosphorylation being one important mechanism. And for example, in terms of Lewy bodies, the most frequent, the most abundant species of alpha-synuclein are phosphorylated forms of alpha-synuclein. Other ways of post-translational modifications include truncation. We know, for example, that shorter pieces of alpha-synuclein may be more toxic than the full-length protein or even other forms and may thereby contribute to the neurodegenerative process. And preclinical studies have indeed shown that this kind of post-translational modification, targeting this post-translational modification, may be of interest. So what here is just an illustration about the alpha-synuclein protein. It's a small protein, 140 amino acids, has a C terminus and an N terminus and a non-amyloid component that forms the non-amyloid component of amyloid plaques in Alzheimer's disease. What do we know about the mechanism of action of immunotherapy? Well, this is illustrated here on the right side. You have two cells that here's one where alpha-synuclein aggregates, illustrated in blue, and we know that in cells where aggregates occur, that these cells may release then alpha-synuclein that can travel through the extracellular space, may be taken up by other cells, neurons and glial cells, and thereby induce the seeding process inside these different cells and lead to the propagation of the synucleinopathy over time. Currently, the current belief is that the immunotherapy is particularly acting on these extracellular cells, and this action may involve clearance by microglial cells. There are currently 5 monoclonal antibodies in development for Parkinson's disease. 2 on the left side, Prasinezumab, Cinpanemab, are in phase 2, and 3 are currently still in phase 1 development. As you can see here, 4 out of 5 are targeting the C terminus. It's only Cinpanemab that is targeting the N terminus. Cinpanemab is a naturally occurring antibody that was identified in humans, and based on this discovery was then generated a recombinant antibody. As you can see here, there is affinity, some affinity for monomeric physiological alpha-synuclein, which means that these antibodies are not entirely specific for the most toxic forms of alpha-synuclein, but there is a larger selectivity to the aggregates for all these different antibodies. Illustrated in red here is the information for Cinpanemab that this compound has been recently discontinued based on the business report 2020 of Biogen, and within this business report was found the information, the phase 2 study, the SPARK trial with BIP054 in Parkinson's disease, did not achieve proof of concept, so the study did not meet the primary or the secondary endpoints, and Biogen has discontinued the development of BIP054 and will apply learnings to future efforts in Parkinson's disease. Beyond these disappointing news, there have been some encouraging signals, announcement of the first results of the PASADENA trial during the virtual MDS Congress last year in September. The PASADENA trial was a randomized controlled phase 2 trial, including 316 patients with a recent diagnosis of Parkinson's disease. There was an equal randomization to placebo, 1.5 gram or 4.5 gram every 4 weeks for 52 weeks. There was no concomitant dopaminergic treatment, or only a MAO-B inhibitor monotherapy was allowed to avoid confounding symptomatic effects. There are no differences in the primary endpoint, which was the change from baseline in total MDS-UPDRS score versus placebo. Total MDS-UPDRS means here the sum of the first, the second, and the third part. The first two parts are reflecting aspects of motor and non-motor aspects of experiences of daily living, and the third part is the motor examination conducted by the neurologist. There were some positive signals in secondary outcomes, as is listed here, especially for the MDS-UPDRS-3, which is the motor examination, but also for the clinical global impression of improvement and the patient global impression of change. When looking at the MDS-UPDRS-3 scores, you can see here there's a difference of 35% when pooling low and high dose in favor of the treatment compared to placebo. This 35% difference translates to 1.88 points of the MDS-UPDRS-3 scale, illustrating that these differences are quite small, but this is known because the progression over 12 months on this scale is quite limited. When looking at the low dose, the difference was 45% in favor of the low dose, translating again to 2.44 difference, and the difference of the high dose was around 25%, translating to a 1.33 point difference. Based on these findings, the company is currently preparing a phase 2b trial, and within this phase 2b trial, there will also be a group on stable L-DOPA therapy. Beyond on these monoclonal antibodies, there is an active immunization pipeline, including epitopes PD-L1A, PD-L3A, and a compound called UB312. As you can see here, these compounds are still in phase 1 development for Parkinson's disease, but also multiple system atrophy, which is another synucleinopathy based on the accumulation of alpha-synuclein mainly in oligonucleotides, in contrast to Parkinson's disease, where alpha-synuclein mainly aggregates and accumulates in neurons. As you can see here, the target is similar to passive immunization, the C-terminus. There have been 3 trials already completed with epitope. 1 is published. I will come to this in the next slide. And one also in a multiple system atrophy already published last year, and 1 currently under review. And what these different trials have assessed beyond safety and tolerability was the capacity to induce antibodies. And as you can see here, there were antibodies induced against the originate peptide, and quite differences between Parkinson's disease and multiple system atrophy that ask the question to what extent potential differences in the immune status may be relevant for the induction of antibodies in these different disorders. When it comes to this first trial in Parkinson's disease, the AFFOA trial that was published last year, you can see here, these were 24 patients initially randomized to receive either low or high dose of PD-L1A for immunization, separated by 4 weeks, and then patients were followed for quite a long time, re-randomized almost after 2 years into low or high dose, and then receiving a boost, and then 1 year later a reboost. The primary objective of that trial was safety and tolerability. There was no signal that would preclude further development. Of course, there were especially some skin reactions, which are classical for active immunization approaches. But the most critical question is to what extent this immunization therapy was able to induce antibodies against the original peptide, but also against alpha-synuclein. And this is illustrated here on the right side, On the left side of this right panel, you have here the 4 immunizations and the titers obtained after the 4th immunization. There was a significant increase in titers against PD-L1A with a reduction over time, and then the boost and the reboost were again able to reboost somehow this immune reaction with an increase, an immune increase of the antibody titers. When it comes to alpha-synuclein, there was also an increase of antibodies directed against alpha-synuclein after these 4 initial immunizations, and then reduction again with a boost and the reboost again, bringing these titers to higher levels. This was observed in 96% of the patients, 22 out of 23. A few patients had also an assessment of antibodies in CSF, and that allowed them to calculate the relative index between serum and CSF, which was 0.3%, which is within the range to what is observed also with passive immunization approaches. In other words, around 0.3% of antibodies are crossing the blood barrier and can act in the brain. So when considering all these different passive and active immunization approaches, so far the clinical trials have shown that they were safe and well tolerated. In terms of efficacy, there has been some disappointing news with the discontinuation of the cinpanemab antibody, but also some encouraging results coming from the PASADENA trial. Regarding the latter, beyond the many questions and considerations for future trials, one important question would be to understand to what extent a longer follow-up, a longer treatment period would allow to obtain larger effects compared to placebo. Thank you very much for your attention, and I'm looking forward to your questions.